PCR, polymerase chain reaction, is one of the most fundamental techniques that's performed in molecular biology and biochemistry. It's really useful because it allows you to make lots and lots of copies of a specific sequence of DNA. You can use this for things like molecular cloning, where you're piecing together the pieces of DNA in different ways. Or you could do things like sequence it, all these various applications where you need lots of copies of a specific sequence of DNA. The way that PCR does this is through a series of cycles where each cycle is going to double the amount of DNA that gets made. And you're making DNA of a specified region. So we have some terms that we need to kind of get out of the way. I'm going to use the word copy, but the more technical term when we talk about this copying is amplification. So in amplification, we're making DNA copies of DNA, and we're making copies of a specific region of DNA, what we call an amplicon, that is present in a template. And so we have some sort of template, which is the DNA containing the sequence that we want copied. This could be a plasmid if you're trying to do some sort of subcloning out of a plasmid, so a circular piece of DNA. It could be genomic DNA if you're trying to amplify a sequence from a genome. Whatever it is that has that sequence you want to copy is going to be your template. And then the region that you want to copy is going to be your amplicon. We can make copies of that specific region using DNA polymerase or DNA Pol, which is an enzyme that's able to make DNA copies of DNA. DNA polymerase can only copy in one direction, from the 5' end of the DNA, which has the phosphate group, to the 3' end, which has that OH. So it can only go in one direction, and it can only start from double-stranded DNA. So we need to make some condition where we have a short region of double-stranded DNA followed by a region of single-stranded DNA with the bases exposed so that DNA polymerase can know what to copy and go forward putting in the complementary bases. In order to tell the DNA polymerase where to start and where to kind of stop, we need to have these short pieces of DNA that are going to provide those platforms. We call these short pieces of DNA primers. And the more technical term for them is oligonucleotides. And we can shorten that to oligos. So you might see them referred to as like DNA oligos or more typically as just primers. You're going to need two primers, one per strand, because DNA polymerase, remember, can only go from the 5' to the 3' direction. DNA has two strands, and so you're going to have one primer per strand. And these are going to kind of end up bookmarking the region that you want copied. So I think it's a little easier to see in kind of visuals. And so if we think about what's going to happen, we're going to have a series of cycles. And these cycles are going to change the temperature. So we're going to get really hot, and then less hot, and then more hot, and then more hot, and then less hot, and then more hot, and then more hot, and then less hot. Basically, we're going to go through these cycles where we're rapidly changing the temperature. Thankfully, there's a machine called a thermal cycler or a thermocycler that is going to be able to rapidly do these temperature changes for us. So we'll prepare our samples in these little tubes, these PCR strips, and then those will go in the machine, and the machine is going to change the temperature rapidly for us, which is great because in the olden days, they had to actually manually move the tube from like water bath to water bath. And so why are we doing all these temperature changes? First, we need to allow the primers to get access to the DNA to bind to. We have our double-stranded DNA template, and we need our primers to be able to bind one per strand. So we need to basically unzip these strands of DNA, and we can do that by giving them energy. By giving the strands heat, heat is going to allow them to kind of wiggle apart from one another. They're going to wiggle apart from one another at those high temperatures. And when we come apart, we call this melting. And so we go to a high temperature, about like 98 degrees Celsius typically, and that's going to separate the strands. Now, at those high temperatures, the primers wouldn't be able to bind because they'd be kind of like wiggling off of them. So we need to lower the temperature a little bit. And so we lower the temperature so that the primers can bind. We call it annealing when these primers are binding. So melting is when the strands come apart, and then annealing is when the primers bind. Now that the primers are bound, we want to let them go to work. We typically raise the temperature a little bit to give them a little more energy in order to do their stuff. These polymerases they typically come from extremophiles that live in really high temperature environments, so they're really, really stable and you can get them hot. And they'll actually work better when they're a little hotter. And so we get them a little hotter so that they can go and do their copying. Every cycle, you're going to have one primer bind per strand. And so one primer, this primer down here, let's look at it. It's going to start and it's going to go, 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 go till it runs out of steam, runs out of time, runs out of stuff to copy, something. It's going to somehow stop, but it's not really going to be a defined end that you want. Now, this other primer, it's going to be on the other strand. It's going to do the same thing, but for the other strands. So at the end of the first cycle, you're going to have something like this. You have the one ends that you want have the ends that you want, but now we basically want to make it so that this piece is going to end here and this piece is going to end here. Thankfully, what's going to happen is now when we melt these again, these new pieces that we made are now going to be able to be used as templates. And now when this primer comes and binds, it's going to define the end of this piece. And when this primer comes and binds, it's going to define the end of this piece. And so we're going to end up with the pieces that are then going to be the length that we want. And so these pieces are going to start with the primer, and then they're going to extend. They're going to extend to the point at which your other primer had started. And so because they're on different strands, what's going to happen is basically those primers are going to be bookending that region that you want copied. And then in the next cycle, what's going to happen is that you're going to have just those regions that you want copied. I mean, you'll still have all these other parts, but those are going to kind of get diluted out because you're going to have so many of the template that you actually want, the region of the template that you actually want. And you just keep doing this, and then in each cycle, you're unzipping these strands, but now these strands are corresponding only to that amplicon that you want. So you're amplifying this amplicon over and over and over again, and you're getting lots and lots of copies of it. So typically you're going to do somewhere between 30 and 40 cycles of this. You want to make sure you have enough cycles to get product, but every cycle you're going to have the risk of introducing mutations and then those mutations getting amplified further. And you also end up running out of your nucleotides and things like that. In terms of these primers, as I mentioned, we can call those oligos, and we're going to need 2 per reaction. We often refer to one of them as the forward and one of them as the reverse. And that's just kind of in the eye of the beholder. So we talk about DNA from like the 5' to the 3' end. And the 5' is referring to you've got this end with the 5' phosphate, and then you've got the 3' end that has that free OH group. And then the DNAs, we talk about it and we write the sequence of it in the terms of the 5' to 3' direction. Because you have double-stranded DNA, sometimes what happens is you have some genes that are coded for by one strand, but then others that are coded for by the other strands. And therefore, forward versus reverse is kind of going to be in the perspective of whether you're thinking about a gene that is coded for by the top strand or it's coded for by the bottom strand, because this would be 5' to 3', and then this would be 5' to 3'. So if we were to flip over the DNA and just think about this was the top strand and this was the bottom strand, then our perspective would be switched. And so that's just kind of a technical note, but we're always going to be writing from the 5' to the 3'. And so we're going to have some sort of sequence, and we want to design a primer that's going to be what we call the reverse complement of that sequence. So when we talk about a complement, that's like A to T and G to C, those are those favorable base pairs that you'll form between the different DNA letters. So every place that you have a G on one strand, you'll have a C on the other strand. Every place you have an A on one strand, you'll have a T on the other strand. When you want to talk about the DNA sequence, and write the DNA sequence, you write from 5' to 3'. But this strand's running from 5 prime to 3 prime in this direction, and this one's going from 5 prime to 3 prime in this direction. So if we want to think about the sequence that would match this sequence, what we need is the reverse complement. So if we had the complement, that would basically just be saying CT, whatever. But that's thinking about it starting at the 3 prime. And we want to write it from the 5 prime direction. So we need the sequence that's going to be this, what we call reverse complement. Thankfully, there are software tools where you basically just highlight the sequence that you want. Like in benching, you can just highlight the sequence that you want, right click, copy the reverse complement. Or you can stick this into various online tools that'll do it for you. So say I want to make a primer that is going to bind to this sequence right here. I can select the sequence, and now this is this in the forward direction, this is the sequence. But I need this reverse complement of it. So I need a sequence that starts with 5 prime is going to have C, C, and then it'll go this way. What I can do is I can right click on this and I can go to either copy special and do reverse complement that way. Or if I go to copy and now I come over here and do the reverse complement, and then it'll copy that to my clipboard. But it's important that you get the reverse complement because that is then going to be able to bind to that site. You have one for your quote unquote forward strand, remembering that the forward is in the eye of the beholder, and then one for the reverse strand. And then those are going to basically both of those would be written in 5 prime to 3 prime direction, and both of those would be reverse complements of the places that you want them to bind. And remember that their purpose is to provide those double-stranded DNA start platforms for DNA polymerase. One of them is going to act kind of like where you want the sequence to stop, and one's going to be kind of where you want the sequence to start. But that's just stop and start is in the eye of the whole beholder as well, because DNA polymerase can only go in the 5 prime to 3 prime direction, and you're specifying the start of where that DNA polymerase will start copying, but it's going to be kind of one of them is going to correspond to it starting at the end of the amplicon, but going in the direction towards the beginning of the amplicon. And then the other one is going to start at the start of your amplicon and go in the direction towards the end of the amplicon. So you just kind of got to wrap your head around things a little and kind of think your way through things. But at the end of the day, what you have is you have these series of cycles. Each cycle, you're going to melt, you're going to unzip the DNA strands. You're going to then anneal, you're going to lower the temperature so the primers can bind. You're going to raise the temperature a little bit so the polymerase can go and it can start making copies from the primer. And then you're going to do this over and over and over again in cycles. In each cycle, you're doubling the amount of DNA you have. What's really cool too is that the DNA is being extended from these primers. And so the primers themselves can actually introduce new things to the sequence. And so you can introduce mutations that way. You can introduce adapter sequences that way, various cool things. And so your reaction is going to look something like this. In each of your tubes, you're going to typically have some sort of master mix. And the master mix is kind of when you have a mixture that has all the generic stuff. And so it's all the shared components of the PCR reaction. So you might be wanting to do multiple PCR reactions, and then each of those might have a different template or they might have different primers. But what they're going to have in common is that all of them are going to need to have some sort of pH buffer that is going to keep the pH steady. It's going to contain salts and magnesium, which the DNA polymerase needs. It's going to contain the DNTPs, the deoxynucleotide triphosphate, so your ATP, your GTP, your CTP, and your TTP. The DNA polymerase is going to need those in order to put them together to make the DNA. Speaking of which, you're going to need that DNA polymerase. And so this is going to be some sort of DNA polymerase from an extremophile typically, and so you might see something like TAC or Q5. We use fusion, and that polymerase is going to be temperature stable so that you can do all those high temperature manipulations. And so you're going to have that master mix, and then you're going to have the template, which has the DNA that you want to be copied. And remember that the region of that DNA template you want to be copied is called your amplicon. And sometimes the template corresponds just to your amplicon, but often the template is going to be something that contains the region that you want. It contains the amplicon, but then it also has other things. The primers are going to tell the DNA polymerase which part of that template you want copied. So they're going to specify kind of the start and the stop of that amplicon. And so in your reaction mixture, you might make a master mix that contains the mixed components that are shared. So I might say if I was doing the same reaction with different templates but the same primers, I could pre-mix these components. Or if I was doing the same reaction with the same template but different primers, then I could pre-mix the ones that were shared. And so you can kind of strategically set up your reactions to minimize the amount of pipetting that you have to do. But at the end of the day, each of your tubes is going to need your buffer, your DNTPs, your primers, your template, your polymerase, and then water to make sure you get everything to the right concentration. Sometimes you'll have a master mix that you buy that actually contains the buffer, the DNTPs, and the polymerase together. And so I used the Fusion master mix in grad school and it worked really well. But then I was trying the Q5 master mix now and we were getting some weird results. So have to figure that out a little. But yeah, those can be a time saver, but they can also cause some problems sometimes. Anyway, now you're going to go and you're going to set up your reaction. So we put the pieces together and now we need to go stick it in the machine. And so the settings of your machine are going to depend on what you want to copy and what your primer's characteristics are and things. So as we discussed, you have these cycles of melt, anneal, extend, melt, unzip the DNA, anneal, allow the primers to bind, extend, allow DNA polymerase to extend those primers, and then they'll extend them until the point at which the other primer kind of started. Before those cycles, you typically have an initial denaturation where you just give it nice and hot for a longer period. And so you might have like a minute at 98 degrees Celsius. Really denature those strands, make sure all the DNA is unzipped, and give the primers their best chance of having a shot at getting in. Then you have shorter melt cycles than the further ones. You're basically unzipping smaller pieces of DNA than you start with your initial template. And so you don't need as long as a melt step in these future cycles. So you have the short melt step, and then that's at that same high temperature. Then you're going to lower the temperature to allow the primers to bind. And then you're going to raise the temperature a little typically. If you have a high TM, we'll talk about this in a second, you might just do 2 steps. And so you just skip the anneal step and go straight to extend, allowing the primers to bind during that extend period. But in most cases, you're going to have kind of 3 temperatures that you're cycling between: the melt temperature, the anneal temperature, and the extend temperature. The melt and the anneal steps are fairly short. The extend step, well, the length of the extend step is going to depend on the speed of your polymerase and the length of the amplicon. So the different polymerases might have different speeds, and so Fusion, they recommend 15 seconds per kilobase. KB standing for kilobase, which is talking about how many nucleotides long the sequence is. And so you want to give the polymerase 15 seconds per kilobase. So if you had a 3 kb product, then you would do 3 times 15 would give you 45 seconds. Typically, the shortest I would want to go would be 30 seconds. Even if the sequence is short enough that you could do less than 30, I wouldn't do less than 30 typically. And if you have multiple things that you're amplifying, I typically set the time to be the longest one. So if I had one that I calculated would need 45 seconds and another one would need 60 seconds, I would go with 60 seconds. And then you do those cycles, and then you have like a final extension where you let it go for 10 minutes at that extension temperature. And then often you're going to set the machine to hold at some temperature. So convention says 4 to 10 degrees Celsius, but it actually isn't needed according to many. And so we've been doing 12 degrees Celsius, and apparently you can just even set it to not do any hold at all. These DNA products are fairly stable, as opposed to things like RNA. DNA is a little more middlemost sturdy. It doesn't have that hydroxyl group that's going to make it less stable. Anyway, what you saw here is I was talking to kind of mentioned how you might need to adjust this anneal temperature. And so we typically start with 68 degrees. That's a good starting point, especially because often we're doing slick, which our primers kind of have extra regions on the end. So the melt temperature that is predicted by software is going to be different because we're it's not taking into account the fact that not all of the sequence is binding. So this melt temperature, this TM, is a measure of how tightly the primers bind to the template. And the higher the TM, the more energy is required, the more heat is required in order to get the primers to come off. So you need to lower the temperature less in order for the primers to bind if your TM is higher. And a higher TM is going to correspond to a tighter binding of that primer to the sequence. And so we can use the predicted TM to determine the optimal anneal temperature. Basically, what's going to happen is that if you're at too low of an anneal temperature, then these DNA primers are going to bind and they're not going to have enough energy to come unstuck. And so they're just going to start copying from where they are. And what you're going to get is you're going to end up with a lot of nonspecific product formation. But if your anneal temperature is too high, well, now the primers can't even bind where you want them to bind because they've got too much energy, so they're just wiggling off. And so then you're not going to get much product, or you might not even see any product at all. So you can go and you can actually change the anneal temperature. And so if I don't see any product form, the first thing I typically do is I'm going to go and I'm going to lower the anneal temperature. And I'm going to lower it and see if I get product formation. Then I can optimize further from there, but I want to see like, is that the problem? So you can test a range of temperatures. And then if I still don't see anything, what I might do is add DMSO. So DMSO is actually going to, especially if you have a GC rich template, it's going to loosen things up and allow the primers to gain access. Because if the problem is that they can't gain access, that's one way that you can help them gain it. And if I still don't see products, then I might do things like go back and check my sequence, do some fresh dilutions, things like this. What happens though if you get a lot of non-specific product formation? Well, now you've got that opposite problem. And so what you want to do is you want to actually increase the anneal temperature. So you can see that as I increase the anneal temperature, I'm getting less of these non-specific product formation. And so you can see that you'll typically have like one band that's going to be the big band, which is hopefully the product that you want. And so you should have calculated beforehand what's the length of your amplicon, because remember you needed that to figure out the time for your extension phase. And so now you know what size you want to look for, and you can compare it to this molecular weight ladder. And so these are DNA pieces with the known sizes that you can then compare to and see, and you hopefully see that this big bright band is the one that you want. And so we want to figure out how we can get rid of these extra bands. And one way that we could do it is often by increasing the anneal temperature. We can also lower the template or the primer concentration. So you can see that if I have a high concentration of my template, I'm getting a lot of non-specific products. But if I have a lower template concentration, I get more of my specific products and less of the non-specific products. So I've been showing an agarose gel electrophoresis, which is basically the gel where you're going to separate the DNA pieces by size. And the bigger fragments are going to be higher up, and the smaller pieces are going to be lower down, because the bigger products are going to travel more slowly. And so when you turn off the power, you turn off the electricity that was driving them through the gel, they're going to get stuck where they are. And where they're stuck where they are is going to be higher up in the gel. And so this is going to be bigger and this is going to be smaller. And your primers are going to be really small, so your primers are typically going to be at the bottom. And if you don't have the primers react, you're going to have basically just the raw primers. But your primers can actually form additional products. They can kind of amplify one another. They can bind to themselves. They can form all sorts of weird stuff. And so you might see some small products that correspond to those primer dimers, primer things like this. And you can also get hairpins where the primer binds to itself and actually prevents it from working. So there are various things that you want to make sure that when you're designing primers, they're not going to do those things. And so there are tools like Amplifix, which I use, and this is going to, you put in your sequence and it'll basically give you a quality score. And then you can see also if you put in your template, it'll tell you if there are places where it's going to bind non-specifically, if there are risks of primer dimers, things like this. And so you can kind of try to ward off these problems before you actually do the PCR by designing your primers that way. Once you get DNA products that you like, then you're typically going to do some sort of PCR purification, some DNA cleanup, often with one of these spin column purification kits. And what it's going to do is it's going to get rid of those proteins that are left over, those salts, all that various stuff that you don't want, leaving you just with your pure DNA. And you can test the purity of that DNA and the concentration of that DNA using UV spectrometry because DNA absorbs UV-260 light. And so you can use something like a nanodrop or a cuvette, and that'll allow you then to determine the concentration as well as the purity. And then hopefully you get DNA bands that look nice, like crisp, like these ones, rather than things with all these non-specific products. So to review, in a PCR mixture, you have that tube of DNA that has your DNA polymerase. It's got the DNA nucleotides. It's got your salts, your magnesium, all that good stuff. It's got the template, which is the DNA you want part of copied. And the part that you want copied is going to be called your amplicon. The amplicon region is defined by primers or oligonucleotides, oligos, DNA oligos, which are short pieces of DNA, typically about 20 nucleotides long, that bookend the part of the template you want copied. You'll have one for your forward strand and one for the reverse strand. And each of those is going to provide a sort of platform for DNA polymerase to get started from. You're going to go through a series of cycles of melt, anneal, extend. Melt, you separate your strands. Anneal, you bind your primers. Extend, you copy the template starting at the primers. You do this over and over and over again and get lots and lots of copies of your DNA. And then you could do things like slick, where you put them together in cool ways and are able to then get bacteria to make a protein of interest for you.